Well, hello everyone, Mrs. Hanson one more time. I'm kind of excited to share. It's chapter 22. This is our last chapter, taking a first step in the last topic of nucleic acids and protein synthesis. I know last chapter we talked at great length about amino acids coming together to form peptides and polypeptides into full-blown proteins. We're going to explore how nucleic acids such as DNA and RNA are responsible for synthesizing certain proteins for cellular metabolism. So let's share our chapter learning goals. In our chapter together, we are going to be able to draw the structures of both a nucleoside and a nucleotide. That's our first journey in this lesson together. A nucleoside with 2 components and a nucleotide with all 3 components. And that actually helps me sometimes when we get there. When I hear nucleotide, I hear the T, and that represents the number 3, starts with the same letter. You'll know what I mean in just a second when we start drawing. We'll be able to put those together to form short segments of nucleic acids called DNA and RNA. DNA, deoxyribose nucleic acid, and RNA, which would be ribonucleic acid. We'll describe the basic features of the DNA double helix. And you remember last chapter when we talked about an alpha helix in terms of a secondary structure of a protein, we'll see that DNA has 2 winding ribbons complementary to one another. We'll outline the main steps of replication. A lot of cellular metabolism in this chapter. Some of it might actually be a little bit of review from biology days. You'll list the 3 types and functions of RNA molecules. You'll be able to explain the process of transcription. So we talk about translation and transcription in this chapter. And you'll be able to represent basic elements of the genetic code. You have a reading assignment component of our chapter as well, doing a little bit of reading about mutation and genetic diseases. You'll find the basic features of the recombinant DNA, and that's used for the polymerase chain reaction and in DNA fingerprinting, a nice application process of our topic. And boy, isn't this a very timely unit. What are those main characteristics of viruses? You can see our sections. I'm going to talk about Section 1 with nucleosides and nucleotides, and we're going to begin with drawing those. We'll put those together to make bigger structures called nucleic acids and study then the DNA and RNA, which are the 2 nucleic acids. Replication and transcription are part of our process as well, along with the translation, turning out proteins through a process called protein synthesis, reading the genetic code to create a specific protein needed for cellular metabolism. Again, this is the 4th chapter in our biochemistry part of our semester. So let's take a quick peek. Why bring up nucleic acids and protein synthesis? It's just a little short reading here. It says, whether you're tall or short, fair-skinned or dark-complexioned, if you're blue-eyed, brown-eyed, green-eyed, whatever you might be, what makes you unique is determined by nucleic acid polymers that reside in your chromosomes of your cell. It's this nucleic acid called DNA that stores all of your genetic information. And while the nucleic acid RNA is responsible for translating the genetic information into proteins, and those proteins are needed by cells for proper function and development. You know what? Even just a minor alteration, a slight mutation in that genetic sequence has enormous side effects on an organism, sometimes resulting in devastating diseases such as sickle cell anemia, cystic fibrosis, just to name 2 of many possibilities. Our chapter, our last one together in our semester, I know that's exciting. It's been a long semester. In chapter 22, we study nucleic acids and how their genetic information is stored and translated into protein synthesis. Are you ready? You have your notepad out? Let's begin. We know that nucleic acids are unbranched polymers composed of repeating monomers called nucleotides. There are 2 types of nucleotides. We're looking at DNA and RNA as examples of our nucleic acids. So nucleic acids are large repeating monomers of nucleotides. These nucleotides have 3 components, and we're going to look at those components in detail in just a moment. So to have one monomer, this is the single monomer called a nucleotide. Repeating patterns of those over and over and over again give us our nucleic acids. We have 2 major types of nucleic acids based on the sugar present in this monomer. If the monomer is a deoxyribose sugar, we have a DNA nucleic acid. If the sugar present is simply ribose, we have an RNA nucleic acid. So DNA actually stands for deoxy. It's missing an oxygen. Ribonucleic acid. This is a 5-carbon sugar. And it's responsible for storing all of the genetic information of an organism and transmits that information from one generation to another. Mitosis and meiosis. Of course, RNA, ribonucleic acid, translates this genetic information found from the DNA into specific proteins. It's reading the specific amino acids to create unique proteins for all cellular functions. Sounds like quite an important job, doesn't it? So as we explore nucleosides and nucleotides, the component known as a nucleotide consists of 3 components, 3 parts. Alrighty, so we must have a monosaccharide, so that's number 1. We must have a sugar, a monosaccharide, or I'll just abbreviate that as a sugar. You'll need a base, and the base we'll know is always a nitrogen-containing base, and we'll study those functions here. And then the 3rd component is a phosphate group, which is a very high energy bond in these monomers. So the nucleotides have 3 components. And remember, these are the monomers that build DNA and RNA, the little individual building blocks. We have a monosaccharide, which if I highlight here, you can see the 5-carbon sugar right here. This is the monosaccharide piece. It is always a 5-carbon sugar. Do you notice when we draw a sugar with that 5-member ring? This was in our carbohydrates chapter a couple chapters ago now, but we drew an oxygen at the top of a house, and then we drew the sugar component. And notice that when we made a 5-membered ring, carbon number 1 is called the anomeric carbon. The anomeric carbon is always 1 clockwise from the oxygen. Here's carbon 2, 3, 4, and then carbon number 5 comes up out of the ring. So this is carbon 5 in a 5-membered sugar, 5-carbon sugar. This carbon number 1 is called the anomeric carbon. 1 clockwise from the oxygen that's in the ring. Notice here, carbon number 1 attaches to the base. We will learn that this is called a glycosidic linkage. Glycosidic linkage. We'll show that again multiple times. Another component of this molecule here, we want to have, let's do yellow. This component here is the base. And so this is the base that contains nitrogen. And you'll notice that that carbon number 1, the anomeric carbon, always connects to the nitrogen in the base. And we'll see that there are a few varieties of bases. But let me highlight specifically, I'll use a light blue right here. This is the nitrogen in the base that connects to the glycosidic linkage. This right here, this linkage is from the anomeric carbon, carbon number 1, to the nitrogen of the base. Now over here we said that this was the 5 end, this is the 1. Carbon 1 attaches to the base, carbon 5 attaches to a phosphate group. Phosphate is a polyatomic ion, PO4, carries a -3 charge. Remember from our good old polyatomic ion days, phosphate. Well, one of those charges is eliminated here with this linkage. This is a high energy linkage. Since this has just one phosphate group, this would be called a monophosphate. One phosphate group here, so it's a monophosphate. We have a nitrogen-containing base. We'll learn how to name these. Coming up soon, we'll learn the name of those bases. We know that the second component here is the sugar, and the sugar is always a 5-carbon sugar. At carbon number 2 in this particular sugar, the carbon number 2 will identify the sugar. If you see an OH present here, it's not in this picture, but if there was an OH present, on carbon number 2, then the name of the sugar is ribose. But if at carbon number 2 lacks an OH, is missing it, then it's going to be called deoxyribose, that it's missing the OH group. And that word said lacks, but maybe I'll put missing. It does not have the OH group on carbon number 2. So that's what I would see in this picture that we just drew. There's no OH group attached here. So therefore, I now know the identity of the sugar would be deoxyribose. The 5-carbon ring, the 5-carbon sugar, the 5th carbon, of course, is up out of the ring. So carbon 1, anomeric. Carbon 2 determines the identity of the sugar. Carbon 3 has an OH. Carbon 4, and then leads up to carbon 5. And at carbon 5, that's where you start to see the attachment of the phosphate groups to create this building block, this repeating monomer over and over and over. This would be a building block of DNA, since the sugar here is deoxyribose. Deoxy is the sugar, we'll learn, that is found in the DNA nucleic acid. If this structure were identical, but contained an OH here at carbon number 2, then this would be a building block of RNA, because the sugar now would be ribonucleic acid. So there's the identity, the big picture of a nucleotide. 3 components: a monosaccharide, which is the fancy name for sugar, and the monosaccharide will be ribose in an RNA, or deoxyribose in a DNA. It's one of those 2 sugars, and you can tell the difference by the absence or presence of the OH group at carbon number 2. You'll need a base. Here's the base component of this structure. We're going to examine 5 possible bases. And yes, we'll learn those structures as well. And the 3rd component of a nucleotide is the phosphate. Since this is just one phosphate, we would call it mono. If it were attached now to another phosphate group, this would now be a diphosphate. And it could even be extended out to a 3rd phosphate, which would now be triphosphate. So you'll hear triphosphate, diphosphate, monophosphate, just indicating how many repeating PO4 groups there are in this nucleotide. Oh my goodness, you've done great work. You are just drawing the nucleotide structure, naming those 3 areas very clearly, and I'm sure yours is much neater than mine is now. So let's continue. It says that DNA molecules contain millions and millions of nucleotides. Nucleotides are the monomers, right? The repeating pattern, the monomers. Nucleotides contain 3 parts, right? The sugar, the base, and the phosphate. Those are the 3 components of our nucleotides. RNA molecules have a few thousand, so those are smaller molecules. Even though those are quite large, they're not as large as DNA. DNA is found in chromosomes in the nucleus, and each chromosome has a different type of DNA. Humans have 23 pairs of chromosomes, which are 46 total, and contain many genes. And that's the portion of a DNA molecule that's read through the process of transcription and translation to create specific proteins. So I read a gene to create the protein. Just the sequencing of the amino acids is created from reading the DNA molecule. All right, so let's examine nucleosides and compare those to nucleotides. Nucleosides have two components of the monomer, whereas nucleotides have all three components of the monomer. The monomer being the building block we just drew. Nucleoside. A nucleoside is composed of the sugar and a base, whereas the nucleotide has the sugar, the base, and the phosphate. That's the only difference between these two terms, is the nucleotide starts to link together with those phosphate groups. So let's examine the nucleoside component, knowing that these are two of the three components we just drew. We're going to focus on the sugar and the bases present in these building blocks. So in monomer called RNA, the sugar, the monosaccharide sugar, is aldopentose. That means that it's a 5-carbon aldehyde, not a ketone, but an aldehyde. And its name is D, just to represent the dextrorotary part of that name. It's called ribose. D is telling me which enantiomer. Not the L, but the D configuration. Alrighty. Now in DNA, the sugar, monosaccharide, is aldopentose 2-deoxyribose. So here's what we want to do. We want to really see the difference here between the sugars. RNA is simply ribose. DNA is deoxyribose. Let's draw these. You notice that you have a 5-membered ring. Start by drawing oxygen at the top of the house, down, back in, and then close the sugar. Carbon number 1 is the anomeric carbon. I'll highlight that in pink. Anomeric carbon. It's always the one clockwise to the oxygen that's in the ring. And I'm mentioning that because this is the site of attachment to the base, the nitrogen-containing base. All right, so there's carbon 1, carbon 2, 3, 4, and now remember carbon 5 comes up out of the ring. So here's carbon 5. Now look, don't forget to place the remaining hydroxyl groups. The hydroxyl groups at carbon 1 point up. There's one attached to carbon 5 as well. And the absence or presence of the hydroxyl group at carbon number 2 will indicate if it's ribose or deoxyribose sugar. Both the DNA and RNA are a 5-membered ring, oxygen at the top of the house and the 5th carbon coming out. This 5-membered ring is the sugar component of our nucleoside, which contains 2 parts. The nucleoside is the sugar and a base. We can identify the sugar based on the presence or absence of the hydroxyl group at carbon number 2. That is the only difference in this structure, is drawing the hydroxyl for ribose sugar at carbon 2 or leaving it off at carbon 2 for the deoxyribose sugar for DNA. Ribose is the sugar found in RNA. Deoxyribose is the sugar found in the DNA let's take a closer look now at those bases. So we said nucleosides have two components. We have the sugar, which is either ribose or deoxyribose, or the second thing we have is a base. There are 5 different bases, 3 of which have just 1 ring in its structure, and the other 2 have 2 rings in the structure. Let's look at the first of these bases, called pyrimidine. Pyrimidine, hear that sounded out? This is the parent compound. It is a 1-ring structure. Now, out of this parent base, the parent compound called pyrimidine, there are 3 derivatives of bases that are found in these nucleic acids. Cytosine, abbreviated with a capital C, uracil, abbreviated with a capital U, and thymine, which is abbreviated with a capital T. All 3 of these are based on the structure of pyrimidine. I want you to take some time to draw each. You have to be able to recognize the bases by name and structure. So here I say, I don't do this often, but I'm going to write, you have to memorize these structures. And you memorize those and the names associated with them. So let's examine cytosine first. Capital C is the abbreviation for this nitrogen-derived base. So the parent, pyrimidine, notice up here, this is called carbon number 1, and you're going to number around. Alrighty. At carbon number 4, you're leading up to an amine group. And at carbon number 2, you have a ketone group. Now remember, this would be the site of attachment to the sugar. The nitrogen is going to attach to the sugar at the anomeric carbon, carbon number 1. If you see a nitrogen-containing base where it has a ketone functional group at carbon 2 and the amine group at carbon 4, that's your cue that that base is going to be called cytosine. Let's examine uracil. Uracil is also a derivative of pyrimidine. Let's number 1, 2, 3, 4, 5, 6. You have a 6-membered ring. At carbon number 4, you now have a ketone as well as at carbon number 2. Alrighty. And then finally, uracil, go over to thymine. Thymine also has the 2 ketones at carbon 2 and carbon 4, but you're adding a methyl group as well. So it has the most functional groups of those 3. You might want to pause the video and really take some time to draw carefully each of the structures. Pyrimidine is the parent. Notice how I'm saying adine as its last name, pyrimidine. Cytosine, uracil, and thymine are derivatives of this parent compound known as pyrimidine. You need to memorize those structures and the name associated with them. Again, this is just a practice skill of draw, draw, draw. Probably flashcards would be a great idea here. All right, so you're back. If you've paused the video to finish drawing, perfect. Let's take a look at the next 2 bases. We said there are 5 possible bases in DNA and RNA. The first 3 of the 5 are single ringed. The next 2 have double rings. Notice here, the purine is the parent compound. It has 2 rings. Carbon number 1, or that's this position 1, 2, 3, 4, and then it comes up 5, 6, and then back over, here's 7, 8, 9. Now, adenine and guanine are two-ringed derivatives of a parent base called purine. What do you see as their differences? I notice here that adenine places an amine group at the top of carbon 6. And guanine has two differences. I can see a ketone functional group at carbon number 6 and an additional amine group. What makes adenine and guanine different from one another is this presence of the NH2 group at carbon 2 in that second ring. This is the site of attachment to the sugar. So this nitrogen will attach to the sugar, alrighty, and that would create a glycosidic linkage to either the ribose or deoxyribose sugar. Adenine and guanine are based on the structure of purine. The parent compound is purine. Its two derivatives are adenine and guanine. Pause the video and take your time drawing those, really focusing on the structure as you do so to help commit those to memory. We'll then pick up, and again, remembering the DNA contains 4 of the 5 bases, and RNA contains only 4 of the 5 bases. What's in common? Adenine, guanine, and cysteine. And what's different? DNA will contain thymine, whereas RNA replaces thymine with uracil. T is replaced by U. So the thymine that is present in the DNA gets replaced by the uracil in RNA. There is no thymine in an RNA. There's no uracil in a DNA. They have 3 bases in common, A, G, C, adenine, guanine, and cytosine. The 4th base is unique. DNA uniquely owns thymine. RNA uniquely owns the uracil. T gets replaced by U. All right, so now we understand that nucleosides have 2 components, right? They have the sugar, either ribose or deoxyribose, and a base, and they have 1 of 5 bases. Nucleotides have 3 components, the sugar, the base, and the phosphate. And this is the monomer then that builds together RNA and DNA nucleic acids. So this is the monomer. This is just a portion of the monomer. It's lacking the phosphate group. When we start building nucleosides, we join together the anomeric carbon. That's just carbon number 1 on the sugar to the nitrogen in the base. And I've mentioned that. The site of attachment is the anomeric carbon, carbon 1. It's the carbon that is just clockwise 1 position from the oxygen in the ring. To name the nucleosides that we're building, the nucleoside, of course, remember, is the sugar and the base. How do we name that? Well, if these bases arrived from the pyrimidine, you'll use the idine name. What does that mean? Idine, I-D-I-N-E, I-D-I-N-A. When it's cytosine is the name of the base. To name that base in the nucleoside, you would call it cytidine, uridine, thymidine. That the original sugar is going to end with the idine name. And again, this is the 1 ring base. Now, to name the nucleoside derived from a purine, which is the 2-ringed base, these are going to receive the ocene suffix. So say that with me, ocene. The adenine is the name of the base. Adenosine is how we would name that in a nucleoside. Guanosine is how we would name that as a nucleoside. So the purine parent gets an ocene suffix. The pyrimidine parent gets the odine suffix. Those are mouthfuls, I know. Now, the other part of the nucleoside name is the presence or absence of the term deoxy. So identify the sugar and decide, do I need to say the term deoxy or leave it off? If it is a deoxyribose sugar, it's lacking the OH at carbon 2, you put a prefix on deoxy. If the sugar has the OH group at carbon 2, you don't hear a word about the sugar in the name. You just ignore that and just name the nucleoside. Let's take a step-by-step practice. Let's draw the 5-membered ring ribose. So again, on your paper, top of the house has an oxygen. It's a 5-membered house, so just draw a 5-membered ring. Carbon number 1 is the anomeric carbon. The anomeric carbon is the site that joins the base to the sugar. Carbon 2 has the hydroxyl group, so I now know that it's ribose. This is the enantiomer, not D, it is D, not L, so that's why you see the enantiomer D. But I know that it's ribose sugar because I can see an OH group present on the second carbon in the ring. Here's carbon 3, carbon 4, carbon 5 is out of the ring, and the sugar will contain an OH at the end. Now remember, if I added a phosphate here, I just turned my nucleoside into a nucleotide. The only difference between the nucleoside and nucleotide structure is the absence or presence of this phosphate group here at the end. Since I'm drawing a nucleoside, I don't show the phosphate linkages. Now we're drawing cytosine. Whoops, I touched the wrong screen. Cytosine is the name of the base. Are you getting familiar with what cytosine looks like? I told you have to draw those so many times that you remember the structure. Here's cytosine. We have a 6-membered ring. We have an amine group and a ketone group coming out of that structure. So that's what we're drawing here once again. Remember, repetition's our friend as we're learning. So each time you draw, you're learning to commit to memory the structures of these bases. The anomeric carbon, carbon number 1, and the nitrogen that's in the base form a glycosidic linkage. You will see, and I'll highlight in yellow, carbon 1 to the nitrogen is a glycosidic linkage. I didn't spell that wrong. And that just simply means I'm connecting a sugar to a base, a glycosidic linkage. I highlighted in yellow, the anomeric carbon. Notice that we have the OH group. So we're going to put this together. We just drew a nucleoside. The sugar itself is ribose. So I don't hear anything about the sugar in the name, and we're just simply going to change the cytosine to cytidine. Remember this slide here. If the base that we're drawing is a 1-ring base, which means it's a pyrimidine derivative, the base is now going to end with adine, and you're naming the nucleoside cytidine. That's it. That's the name of the nucleoside, cytidine. Now, if, and it didn't, but if the OH group was absent, let's say if it's not there, what would it now be called? Well, it would be called deoxy citadine. There. The presence or absence of the OH group at carbon 2, if it's present, you just simply name citadine. If it is absent, you would name it deoxycitadine. And we are naming nucleosides, 2 of the 3 components in the building block of nucleic acid monomers, hooking together the 5-carbon sugar with a base. The base in this example was cytosine. Cytosine is from the parent. Remember, I'll just keep flashing these over and over. Cytosine is from the parent pyrimidine, which means we change the ending of the base from cytosine to cytidine to recognize the parent name. We just named the parent base here, cytosine, when it's hooked to the sugar as cytidine. Cytidine is the name of that nucleoside. These are just big words. I keep saying them over and over so they start to sound familiar. Should we try another? Let's hook together in this next structure deoxyribose and adenine joining to create deoxyadenosine. That is a deoxyribonucleoside. And then we're going to highlight or color in the glycosidic linkage. All right, so when I hear deoxyribose, we draw a 5-carbon sugar, oxygen at the top, carbon number 1, 2, 3, 4, 5. Carbon number 1 is called the anomeric carbon. The anomeric carbon is the site of attachment between the base and the sugar. Now, the base attaches to this nitrogen group right here, and that's going to attach directly to the anomeric carbon, which is 1 clockwise from the oxygen in the ring of your sugar. Notice at carbon number 2, we're lacking the oxygen. So now I know that it's deoxyribose. Now, how do we know how to draw adenine? Well, that's the part I'm challenging you to study. You have to know, okay, adenine is one of those 2 bases that has 2 rings. Guanine and adenine, A and G, have 2 rings. Its parent is purine. And we said when we are naming these, the purine as a parent gets named with the suffix ocene. We wrote that on this slide, ocene. So in the bases would be named, let me go here. The bases were named adenine and guanine, but if we're naming those bases as part of a nucleoside, you hear them stated as adenosine or guanosine. And then depending on the sugar, if the sugar has no oxygen at carbon 2, you'd hear the prefix deoxyadenosine. And that's really what we were just drawing right here, wasn't it? Deoxyadenosine. Carbon number 2 lacks the sugar, so I know the sugar is deoxy. And I know that the base is adenine, which is a 2-carbon, 2-ringed parent. So we hear it named adenosine. The name of this nucleoside is deoxyadenosine. The anomeric carbon, carbon number 1, attaches to the nitrogen in the base. I'll highlight this linkage as the glycosidic linkage between the anomeric carbon and the base. Now, this is a nucleoside because it doesn't have the phosphate groups coming off here. If we were going to draw a nucleotide where all 3 components are present, this is the site where I would start adding the phosphate groups at carbon number 5 in our sugar. All right, let's really practice. We have 3 practices, and I kind of put them all on the same slide, but I'm going to go through each one separately. Name the nucleoside. Well, here's the step-by-step process. Step number 1, you want to identify the base. So I go in and I try to find the name of the base. Go in and locate these bases and just name them. What are those names? Step number 2, we want to decide, do we have a deoxy or just simply a ribose sugar? So I want to examine carbon 2. I want to identify the sugar. Is it deoxy or not? And then we simply assemble the names together. You can quickly see the importance of memorizing the structures of the bases when you're asked to name these structures. Give yourself time, pause the video, and really work out to commit to memory the identity of these bases. Come back to me when you've written out the names of the bases, identified the sugar, and put the name together, kind of assemble the pieces, and we'll check our work. Give yourself think time, my friends. All right, welcome back. Let's go through these step by step. Let's examine the first example. We had to identify the base. When I said identify the base, this is that portion of the nucleoside. I know that it's one ring. So I really narrowed it to 3 choices. Is this cytosine, uracil, or thymine? Well, one of the things that I can help you with is when you notice that there is no OH group here, you've already decided that this is deoxyribose sugar. And that tells me that that is a building block of DNA. And I know that A, G, C, and T are only found in DNA. That helps me eliminate a choice, doesn't it? I know that it cannot be uracil because uracil is not one of the choices for DNA. So now I have it narrowed down to cytosine or thymine. Which one is it? Well, you can see that it has this methyl group right here, which makes this thymine. So step number 1, you've identified the base. Remember that thymine, which has the parent of a pyrimidine, is going to end with a new suffix. The suffix is -ine. The sugar deoxy and the base thymine. You want to start with the deoxy. You want to write out thymidine, and you've assembled the pieces of this nucleoside name. So you're going to put it together. Identified the base. We said it was, you got it. We said it was thymine. You identified the sugar. You got it. It was deoxyribose. And when you put those together, you put the name as deoxythymidine. Deoxythymidine. The base, the sugar, and putting it together. Now, just as a reminder, if it had an OH here, then it's not here, the deoxy part. All right, so the prefix is only given to the sugar if there's no oxygen present at carbon number 2. We know it's a deoxyribonucleoside. Nucleoside contains 2 components, the sugar and the base. And this linkage right here is called a glycosidic linkage, hooking together the anomeric carbon to the nitrogen in that ring of the base. How about those 2? Did you get that far? Identify the base, identify the sugar, and put the pieces together to name these nucleosides. Step number 1, did you identify the sugar? In each of these components, I have to start by identifying the base and the sugar. Presence or absence of an OH. Those are the 2 key components of naming the nucleosides. Find the name of the base, identify the sugar. All right, so here's kind of the step-by-step process. Notice in your first example, there was just one ring. And in one ring, you had three choices. Is it cytosine, uracil, or thymine? Well, one strategy I'm recognizing is that the sugar itself had an OH group, right? So I know that the sugar is ribose. And that tells me it's a building block in RNA. And therefore, I know that thymine cannot be a choice because it's only found in DNA. So that narrowed the two choices to cytosine or uracil. Which one is it? Well, the key here is that the top functional group matches uracil. Cytosine would have an amine group up there, whereas the uracil has a ketone. So if the base here is deriving from pyrimidine, you're going to change its name into an adine, right? And that I see that the sugar itself is ribose. It's got the OH group. So we don't hear deoxy in there. We just strictly say uridine. The name of this nucleoside is called uridine. It came from the base uracil, and I don't hear anything about the sugar because it has the OH, the hydroxyl group there. So just simply call this uridine. And you had a third example to work through. This third example, notice that you had 2 rings in the base. That told you to go and look at your purine derivatives. That gave you 2 choices. Is this adenine or is this guanosine? Step 1, name the base. Notice that it's a derivative of purine, so it's going to end in an ocene name. The suffix here is ocene. Which one is it? Well, you'll notice here that it has the OH group, and that's got to be here. The ketone functional group is in the guanine structure. So guanosine. But it's missing the OH here as well, so we have to say that as part of its name. We hear the prefix deoxy and the suffix ocene. So this is called deoxyguanosine. The 2-component monomer. This would be found in DNA as well. DNA because it's deoxy sugar. And the name of it is just deoxyguanosine. Nucleosides and nucleotides. All right, let's keep going. Just really adding that third component into this structure, which would be the phosphate group. So nucleotides, hear that T there? The nucleotides are formed by adding a phosphate group to the 5' hydroxyl of a nucleoside. So let's take a look at this structure here. We have cytidine. Notice that I don't hear deoxy, so I know that the OH is present on carbon 2 of the sugar. At carbon number 1 is the glycosidic linkage that leads to the base. What's the name of this base? Oops, I wanted to highlight. What's the name of this base here? Are you getting to where you can recognize by structure the names of these bases? The name of the base is cytosine. To name it in a glycosidic linkage, we'll call it cytidine. Cytidine, hear this word, ribonucleoside. The nucleoside consists of 2 components, the sugar plus the base. Now when we add the phosphate to carbon 5 of the sugar, you're turning your nucleoside into a nucleotide. That's the only difference in this structure. Locate carbon number 5 of your sugar. You're going to remove the OH group and you're going to add the phosphate. Remove the OH and add a phosphate over there. So this cytidine, which was the nucleoside, gets a third component, and that's the phosphate, the third part of a nucleotide. Now, this phosphate group has a very high energy bond here as well. We'll learn that each time I add a phosphate, it's a high energy bond. Since this has just one phosphate, the prefix for one would be mono. So this is a monophosphate nucleotide. Now, C stands for cytidine. M stands for mono, and P stands for phosphate. Its three-letter code would be CMP, standing for cytidine monophosphate. At 5' monophosphate is abbreviated CMP. What does this 5' mean? It means at carbon 5, we added a phosphate group, the carbon that's up and out of the ring. Cytidine 5 monophosphate is the name of the ribo. Why do I know it's ribo? Well, because I can see the OH group. It's not deoxyribo, it's just strictly ribonucleotide. Let me emphasize this again. See how a nucleoside converts to a nucleotide by adding a phosphate. And we add the phosphate at carbon 5 of the sugar. Now, just to say, what if I added another phosphate? I can do that. I would have diphosphate, and my abbreviation would be CDP, cytidine diphosphate. And I could have CTP, cytidine triphosphate. So that middle initial tells me how many phosphate groups are attached. Mono, di, or tri, indicating how many phosphate groups are attached to this 5th carbon. Okay, lots of good drawing here, isn't it? All right, we did cytidine 5 monophosphate. Let's try another. Let's turn this nucleotide into deoxyadenosine 5 monophosphate. All right, so first of all, let's break this down slowly. The deoxyribonucleoside. That means the nucleoside has two components. The two components are a sugar and a base. What's the sugar? Well, the sugar is deoxyribose. So when I draw that sugar, I know that it's a 5-membered ring. Carbon 5 comes up and out. And when I draw that, the anomeric carbon, carbon 1, is going to lead up to the base. Now it says deoxy, so carbon 2 will not have an OH, but carbon 3 certainly will. Alrighty. So there's my structure I'm learning to draw over and over again, called the deoxyribose sugar. The base, adenosine. I've got to go in and remember how to draw adenine. Now I know this isn't on your paper, it's in your brain. I have to remember how to draw adenine. I know it's adenine because I see 2 rings and I see this ammonia, this amide group up here, this NH2, adenine. So we have the glycosidic linkage, which attaches the anomeric carbon, carbon number 1, to the nitrogen in the base. The base is adenine. When I name it as a nucleoside, I hear the deoxy part because it's lacking the oxygen at carbon 2. Deoxyadenosine. Now I want to turn that in from a nucleoside to a nucleotide. So I'm going to add the phosphate group. At carbon number 5, we add 1 phosphate. So that's the mono part of the prefix. Look at this 4-letter code now. The little d stands for deoxy. So if my sugar is lacking the hydroxyl group at carbon 2, you hear deoxy. The A stands for adenosine monophosphate. Adenosine monophosphate. The little D out front to let us know that there is no hydroxyl group at carbon 2. So this would be a nucleotide building up DNA molecules, DNA for the deoxyribose sugar. You have to be able to draw these structures or pick them out. I mean, if it's a multiple choice assessment, you have to be able to name the base from looking at its picture. That's the skill we're working towards. So deoxy adenosine 5 monophosphate is abbreviated as DAMP. Keep in mind, if I added another phosphate, it would be ADP for diphosphate. And if I added a third phosphate, I could have ATP, deoxyadenosine triphosphate, and so forth. So the second letter of this code tells me how many phosphate groups there are, mono, di, or triphosphates. ADP is an example of a diphosphate. Let's draw that structure together. So you just drew AMP. Draw it again, adding a second phosphate group, and now you've abbreviated this as ADP. Notice the difference. I said that we just drew it, except we drew a deoxyribose sugar, and now we're adding on that oxy. So this is adenosine 5' diphosphate. Notice that I have 1, 2 phosphate groups. Adenosine comes from the adenine functional group here. That matches the name of the base. Adenosine 5' diphosphate. This would be a nucleotide. Oops. And that would be in RNA because the oxygen is present. If the OH group is lacking at carbon 2, it's a building block of DNA. If the OH group is present at carbon 2, those are building blocks of RNA. So 3 components of the nucleotide is the sugar, the base, and the phosphate. The 3-letter or 4-letter codes tell us the sugar. The sugar is told to us by the absence or presence of the little d, right? D, ADP, would be deoxyadenosine 5 diphosphate. Just straight old ADP, I know the sugar now has the hydroxyl group, so we don't hear a prefix. So you just write ADP, and now you know the sugar is ribose. Whew, let's keep practicing. Here's an example of a very familiar molecule, ATP, adenosine triphosphate. So now you're just redrawing that structure one more time, emphasizing what that middle code stands for. I have 1, 2, 3 functional groups of phosphates here, right? And that right here means triphosphate. You know that the base comes from adenine, that's this portion right here, and that the sugar is ribose because carbon 2 has the presence of the OH. Here is the anomeric carbon, the carbon 1 spot to the right clockwise of the oxygen. And the anomeric carbon attaches to the nitrogen in the base, creating what we call a glycosidic linkage. At carbon number 5, you have the phosphate groups attached, and those are the high energy bonds. Each time you break one of these bonds, you're releasing a high amount of energy for cellular metabolism. ATP, adenosine triphosphate. All right, if you need time to finish drawing, pause the video. You've got some really good memory work to do here, just some really, if it were me, my recommendation is flashcards to learn the 5 bases. Let's summarize. The term nucleoside means that it's composed of 2 parts, the sugar and the base. A nucleoside is composed of a monosaccharide attached to a base from the anomeric carbon to the nitrogen of the base. A ribonucleoside is the building blocks of ribose, and deoxyribose has the building block of deoxyribose sugar. So the absence or presence of an OH group at carbon 2 determines if I have ribose or deoxyribose. A nucleotide composes 3 parts. You have a monosaccharide, the base, and the phosphate. Now, a nucleotide can be it could be a mono or a di or a tri in that 3-letter or 4-letter code, mono, di, or tri. How many phosphates are attached to carbon number 5 from the sugar? A DNA molecule contains deoxyribonucleotides as its building blocks. And in there, you're going to find A, G, T, and C as your 4 bases. And we know in RNA, T gets replaced with U. We had said there's 3 bases in common, A, G, and C. But in DNA, there's thymine, which is unique to DNA. And in RNA, there's uracil, which is unique in RNA. We've also highlighted some interesting things about DNA versus RNA. We said that the DNA has 4 choices of base: adenine, guanine, cytosine, or thymine. We can see that the abbreviation for adenine is a capital A. The name of the nucleoside would be deoxyadenosine. Make that light yellow. A turns into deoxyadenosine, and to name it as a nucleotide with just 1 phosphate, it becomes deoxyadenosine 5-monophosphate, abbreviated as little d AMP, deoxyadenosine 5'-monophosphate. Coming from this base, adenine. In DNA, there's a base called guanine. It's abbreviated with a capital G. Its nucleoside would be named deoxyguanosine. Its nucleotide, deoxyguanosine 5-monophosphate for just 1 phosphate group. Guanine comes from this derivative, another 2-membered ring. And of course, it's abbreviated DGMP, deoxyguanosine 5'-monophosphate. Cytosine is the 3rd base found in DNA, abbreviated capital C. Its nucleoside would be named deoxycytidine, and therefore its nucleotide becomes deoxycytidine 5'-monophosphate, little d CMP. That's coming from cytosine. And the 4th base found in DNA is thymine. And thymine has a capital T abbreviation. Deoxythymidine is the name of the nucleoside. And if you add a phosphate, you would name the nucleotide deoxythymidine 5'-monophosphate, DTMP. And that right here is your thymine base. And all of that remains the same for an RNA, except now we see, of course, that uracil is replacing thymine. And you do not have the little d any longer. So the little d is meaning that this is deoxyribose. And that's why you see the little d out front for the deoxy part. You do not see that for the ribose sugar. So RNA is strictly AMP, GMP, CMP, UMP, adenosine, guanosine, cytidine, uridine. The names of those nucleosides. And to put it into a nucleotide, you rewrite everything as it is, but leave off deoxy up above, right? Adenosine 5-monophosphate, guanosine 5-monophosphate, cytidine 5-monophosphate, uridine 5-monophosphate. Take some time with this slide, pausing the video, and really thoroughly understand what you're writing. Take a moment to process as you write. All right, let's put some practice in before we bring closure to our first lesson. Let's practice. There's been a lot of new words coming our way. Let's identify the base and the monosaccharide in each nucleotide and then name it. Name the nucleotide and give its 3 or 4 letter abbreviation. So I'm asking you really to name the nucleotide, this column. Which one is it? So the first step, of course, identify the base. Second, identify the sugar. And 3, assemble the pieces. What's the name of this base? What is it? What's the name of this sugar? What is it? And part of this also will be to notice that there's just one phosphate group, not 2, not 3, but just one, so I know it will be a monophosphate. Well, right here, I want you to notice that as soon as I notice the sugar was ribose, I know that I'm in a building block of RNA. So now my choices are A, C, G, or U. What is that base? If you have to keep peeking, here are your pictures again. Did you find an answer? Let's go through it. When you identified the base, it came out as uracil. When you identified the sugar, it came out as ribose. The name of this base is uracil. The name of the sugar, ribose. Put it together and you come up with uridine 5-monophosphate. UMP is its 3-letter code. Uracil becomes uridine. 5' tells me that this site of attachment is at carbon number 5 to that phosphate group. This right here is the glycosidic linkage between the sugar and the base called uracil. Uridine 5-monophosphate. And remember, if there was another phosphate here, you'd see another attachment of POO, then it would be diphosphate. If there was a third, it would be triphosphate. But here we only had one, so we knew it was a monophosphate. Shall you try another? Identify the base, identify the monosaccharide, and name the nucleotide, giving me its 3 or 4 letter abbreviation. Now let's think. Give yourself pause. Think it through. Pause the video. Come back when you're done thinking it through. All right, let's process together. We said the first skill is identifying the base. What is the name of that base? Well, it matched thymine. What is the name of the sugar? Well, the sugar is deoxy. It does not have an OH there. So what does that sound like? And also notice, just to really emphasize, it's monophosphate because there's just one phosphate there. There could be another, and there could be a third. It could be monophosphate, diphosphate, or triphosphate. So with this example, we found the base to be thymine. And the sugar had to be DNA sugar because it was deoxyribose. So in a DNA molecule, when the base is thymine, the name of the nucleotide is deoxythymidine, 5' monophosphate. It is going to be abbreviated with a 4-letter code, D, TMP, T standing for thymine, M standing for mono, and P standing for phosphate. Deoxythymidine 5' monophosphate. You've named the nucleotide. Remember, if I did not have any phosphate, we would name it as a nucleoside, but I'm naming it as a nucleotide when all three components are there. Let's really practice now. Draw the structure of GMP. Draw it. Well, here's some guidance. What does GMP stand for? Translate the abbreviation to a name. Then draw the sugar. Then draw the base. Connect the base to the anomeric carbon from the nitrogen in the base. And then add on that phosphate group at the 5' end of the sugar. Pause the video. Give yourself the opportunity to sketch it out. Don't just wait for me to give you the answer. Try it out first. Pause, think, and come back. Are you ready to check? Well, first of all, GMP, guanosine, 5' monophosphate, guanosine. That's the G. I bet you knew that from guanine. Guanine is this structure. I have to know that. Guanine is a 2-ringed base, and the sugar will contain the OH group, right? So start by drawing the sugar. The sugar will contain a 5-membered ring where carbon number 5 is up and out of the ring. The anomeric carbon is going to attach to the base. I don't have a little D here for deoxy, so carbon number 2 has the OH group. So there's the first step, is just drawing the base, draw the sugar first. Draw the sugar. Now let's draw that base, and here's the site of attachment. So it's going to lead up to this nitrogen that's in a ring. And I'm just drawing that structure as I see it. There's that base. I need a double bond there, double bond there. So I drew in the base, attached it as a glycosidic linkage between the anomeric carbon, carbon 1, and the nitrogen that's in the base called guanine. Right? Now what? That would be guanosine. That would be a nucleoside, but I want to turn it into a nucleotide, and I want to add 1 phosphate group. And we do that right here at the carbon 5 position. So that's going to lead to an oxygen. And there's your phosphate, monophosphate. PO4 is phosphate. Let me show that kind of clean. So as you're putting those together, you drew the sugar first, then you drew the base, connecting the nitrogen to the carbon 1 of the ribose. That's the anomeric carbon. And this was your final structure. We drew guanosine. We drew the hydroxyl group, and we placed the phosphate to create guanosine 5' monophosphate. I can't emphasize enough how important it is to practice these skills. And let's take a pause. We'll come back, give ourselves a little rest. We've been talking for a long time, over an hour already, practicing section 1. When you come back, we're going to move on and look at nucleic acids, DNA, and RNA, and start looking at their structures. So good work so far. Take a break. Come back for lesson 2 when ready.